This video will walk us through how to add a new marker-based segmentation to our chip cytometry data. In the case of tissue, we use a marker to denote single cells. This is in contrast to the procedure for PBMCs, where we use transmitted light to segment individual cells. For marking cells, we recommend a nuclear stain such as DAPI, HOIS, or propydium iodide. First, we're going to use the info panel, and we're going to find the cell recognition tab. This tab will tell us right away what the active segmentation is currently, and then also what marker that segmentation was run on. In this case, it was a HOIS stain. If I want to run a new segmentation, I just have to order new segmentation. Before that, though, I'm going to choose the test mode option. The test mode option will run the segmentation only on one position. So this is very nice if you're trying to fine-tune the segmentation algorithm for your data, because you don't have to wait for the entire chip to run. It's just a single position. With that option selected, I will select Order New Segmentation. I'll then select all cell types, single or multiple markers. It'll then ask me what marker I would like to base the segmentation on. I'll choose HOIS again, click OK. It'll now ask me what sensitivity I'd like to use. 500 is the default value. That's a very good place to start. Lower numbers will indicate lower sensitivity, which will make the algorithm find fewer cells. Higher sensitivity, higher number, will make the algorithm find more cells. Click OK. It'll then ask me if I want to execute this job manually. Typically, you do want to do this. This will add the job to the queue, but won't run it immediately. I'll then go to the bottom of my queue here, and I can right-click, and then one of my options is to execute this job. I'll go ahead and execute the job. I will be warned that resetting the segmentation to plan will delete all gates and reset all value calculations to planned, and ask if I want to proceed. This is essentially saying that this is going to overwrite the old segmentation, and this will also delete all the gates and all of the fluorescence value calculations that were based on that old segmentation. Usually this is just fine. In this case, I'd like to keep my old segmentation, so I'm going to click No. The last part of this process is to recalculate the fluorescence calculations for each marker using the new segmentation. So let's assume that I picked yes, I did want to use the new segmentation. The final step here would be to go to any marker, click the double caret, and from this drop-down pick, recalculate fluorescence for all markers. We're then asked if we'd like to execute this job immediately or in the background later. I'll pick immediately by clicking yes. And you can see we're now processing and calculating new fluorescence values using this new segmentation for each position.